Okay, good afternoon, everybody. Thank you for taking time for our next INTERMICRO seminar, which is kindly given by Dagmar Šrůtková from our partner workplace in Nový Hrádek from the Laboratory of Biology, and who will be talking about mice and immunity and microbiomes. I mean, I have a lot of questions because I probably didn't understand a lot of things, but I'm happy to welcome a lot of people online, but also a couple of people in-house. So the floor is yours, and we're looking forward to your presentation.
Thank you, Honza, for a nice introduction. Thank you, all the ladies, gentlemen, friends, to coming here and hear me. Today, I would like to introduce you our study that was published in January of this year, and that was accepted to publication directly on a New Year Eve. And this study was mainly done by our PhD students, Tereska Horníková and Anička Jelínková, who performed microbiome analysis for us. And we are happy that today we have approximate 500 downloads of this paper. No citation yet. I think that I don't need to introduce our laboratory that is surrounding in that is located in surrounding of Eagle Mountains, close to the Czech-Polish border. And in our laboratory, we are investigating the role of microbiome on the physiology and immune response development in healthy and disease conditions. To that studies, we use our germ-free models, animal models that are germ-free piglets and germ-free mice. To study the mechanism of allergy, we previously established the model of airway allergic inflammation and also food allergy development induced by systemic sensitization through epicutaneous through intraperitoneal route. And nowadays, newly, as you will see today, we establish a model of induction food allergy through epicutaneous sensitization through disrupted skin barrier, which occurs in approximately 40% of atopic dermatitis patients. And this process is connected to so-called atopic march, where occurs atopic dermatitis and food allergy development in the same time. To our study, we have a driving hypothesis called dual allergen exposure hypothesis that says that in healthy individuals, if the first or early application of or contact with food allergen goes through oral route, then is induced oral tolerance to food allergen, and no allergy is developed. But in predisposed individuals, which is mainly which could occur in atopic dermatitis patients, where the individuals has disrupted skin barrier, the first application of food allergen on the disrupted skin could lead to sensitization to this food allergen, and then development of food allergy in the Th2 pathway with induction of IgE antibodies and mast cell response. Our study has in our study we have the aim to determine the factors, like genetic predispositions to immune response induction or microbiome of the gut and skin, how these factors could lead to development of food allergy. First, we determine how the genetic background of the experimental mice could influence the development of our model. We used commonly used bulb C and black 6 mice. That was females, 2 months old, and we shaved them the hairs on the skin. Then we striped their shaved skin by tape to disrupt the skin barrier, and after that we applied patch soaked with ovalbumin, with allergen ovalbumin, for 7 days. This phase was repeated 3 times. And 10 days after the third epicutaneous sensitization, we applied 6 doses of allergen ovalbumin intragastrically. At the last of application, at the 6th day of 6 dose application, we took the samples and analyzed the immune response and also the microbiome. We observed when we observed in sera of mice that mainly in bulb C mice show elevated level of ovalbumin specific IgE and also IgG1 antibodies that are antibodies typical for allergy reactions. Also, we observed that bulb C mice has highly elevated level of Th2 related cytokines like IL-4, IL-5, IL-6 or IL-10 in spleen cell cultures. Interestingly, we didn't observe any Th1 cytokines. Here I showed the interferon gamma. At the end of the experiment, we also measured anaphylactic hypothermia. That is measured like a drop of core body temperature by a thermal by rectal thermometer 15 minutes after the application of ovalbumin. And we saw that mainly bulb C mice that treated by OVA displayed big drop of the rectal temperature. And also this group of mice displayed from the second dose of ovalbumin application the presence of liquid diarrhea, as you can see on the picture. We also determined the mast cell response, as given that mast cells are a hallmark of food allergy, and we showed that bulb C mice in bulb C mice was visible huge infiltration of these cells into the yeunal tissue. And we determine also mast cell related immune response. And again, we observed that that was again observed mainly in bulb C of treated mice, and that is mast cell protease factor in jejunum and in sera, and also the site-specific cytokines IL-4 and IL-13 in jejunum. And we determine also the factors that are related to the release of histamine and leukotriene from mast cells, and it is, that's arachidonate lipooxygenase and histamine deacetylase. That is these 2 factors. At the end of the experiment, we also determine how the process of epicutanaw sensitization and food allergy induction influence the composition of the microbiome in the intestine and also on the skin. In the intestine, in the feces, we determined that bulb C mice treated by ovalbumin displayed elevated alpha diversity indexes, here presented as Shannon diversity index, CHA1, and observed ASV, and also Fates diversity index. By beta diversity indexing using unweighted unifrac analysis and Permanova analysis, we clearly clustered, there was clearly clustered the microbiota according to OVA treatment. As you can see, the dependency of OVA treatment of the mice, and also the dependency of the used strain on the clustering of microbiome. Further, we determine the abundance of specific bacterial taxa, and we show that the mice has displayed or has highly abundant the taxa that are connected to short-chain fatty acid production, as that are muribaculace, lachnospirace, or prevotelace. And directly these taxa were downregulated, were less abundant in bulb C mice compared to black 6 mice or compared to PBS controls. And that was interesting that we didn't observe these differences in case of skin microbiome in our experimental mice. And beta diversity shows that the dependency only the used mouse strain. Interestingly, we also observed on the skin mainly taxa, the huge abundance of the taxa that are naturally occurring in intestine, that could be caused by contamination from beddings of the co-environment. When we look at the taxa commonly found on skin, that is Streptococcus or Aerococcus and Hydrobacter, there were no differences between among the experimental groups. To the request of reviewer, we performed a multivariate analysis that investigated the relationship between the microbiota abundance and other main features of the immune response to food allergen, as mast cells response and also the cytokine parameters. And that was clearly shown that groups of bulb C over-treated mice clearly cluster together away from another experimental groups. Then we determine how the microbiome contributes to the process of epicutanaw sensitization and food allergy development. To that, we used our germ-free mice read in isolator, that was bulb C mice, and their SPF counterparts. We process again the process of epicutanaw sensitization and also 6 allergen challenge, and then we sacrifice the mice to take the samples. Interestingly, we didn't observe any differences between of a specific IgE level among SPF and germ-free mice, as you can see here. And even also, we didn't observe differences in IgG1. but we saw differences in Th2 related cytokines like IL-4, IL-5, IL-10 or 13, and there was upregulation of these cytokines mainly in spleen cell cultures from germ-free mice treated by OVA. Both groups of mice induce anaphylactic hypothermia, here measured as a drop in core body temperature, but the germ-free but in case of germ-free was this drop more pronounced. And interestingly, germ-free mice didn't induce any liquid diarrhea during the course of albumin gavage. That is only by SPF mice. And our explanation is maybe due to germ-free physiology of intestine and its motility of germ-free mice. We also checked the mast cells response, and we saw that germ-free mice induce higher level of mast cells in jejunum tissue, but we saw no differences in mast cells on the skin of these both groups of mice. And to that, we also saw the mast cells related immune response in jejunum, sera, and also here are the factors for release of histamine and leukotriene. From this study, we can conclude that the importance of careful selection of mouse strains is when we conduct the experiments, not only with mast cells experimental model, but also another experimental model. And besides the genetic background, also microbiome could affect the immune response of mast cells and food allergy development that follow the natural antigen adjuvant-free sensitization through the disrupted skin. And this clearly describe the skin-gut axis connection. I would like to thank my colleague from Nový Hrádek, mainly Tereska Horníková and Anička Jelinková, who did the study, Martin Schwarzer and our laboratory assistant to their help, and also Zuzka Jirásková-Zákostelská for sponsoring our study. That was sponsored by Czech Health Research Council, Štěpán Coufal, who did the great multivariate analysis, and also another persons who provide the mentoring of this project. Thank you for your attention.